Dear students, in this session we are going to discuss about the last disorder under genetic disorder, that is Mendelian disorder, and that disorder is thalassemia. So what is thalassemia? Thalassemia is a autosomal recessive blood disease which appears in children of two unaffected carriers, or we can tell heterozygous parents. So in other words, thalassemia is a autosomal recessive disorder concerned with hemoglobin, so seen in children whose parents are carriers. They don't have disorder, but they have got one gene which is present in them and which is transferred to their children. So since the child receives an autosomal recessive gene from both the parents, chances of child getting this disorder is more. Or you can define this in another way. It's an autosomal linked recessive blood disease transmitted from parents to the offspring when both the parents are unaffected carriers for the gene. That's what I told you. Phenotypically, parents don't have this disorder, but they have one gene which is known to be present in them in recessive form. So if both the parents are carriers and if they have children, chances of children getting thalassemia is more common. So now let us come for the history of thalassemia. So this thalassemia was first reported by Cooley in 1925, and the term thalassemia was coined by Whipple and Bradford in 1932 after this disorder was discovered in Mediterranean region. Now let us see the occurrence. This disorder occurs in different parts of the world, that is Middle East, Indian subcontinent and Southeast Africa. What is the main cause for this disease? This thalassemia is known to occur due to mutation or deletion of genes controlling the formation of hemoglobin chain. That is, there are two major types, alpha and beta of hemoglobin. You know, hemoglobin contains two alpha chains and two beta chains. So therefore, in case if there is mutation in gene responsible for producing this alpha and beta chains of hemoglobin, then this can say lead for thalassemia. So what is the major effect? So because the gene has undergone mutation or the gene has been lost, this leads in abnormal or imbalanced synthesis of hemoglobin chains. That is, alpha and beta chains are not properly synthesized. This leads for anemia, and which is the major characteristic feature of this disease. Thalassemia, let us come for types. See, basically your book tells about thalassemia is divided into two major types. But thalassemia is of three types. What are they? Alpha thalassemia, beta thalassemia and delta thalassemia. So let us come for what is this alpha thalassemia. So in case of alpha thalassemia, this is caused due to decreased alpha globin production. That is, the alpha chain which is produced, they have got few amount of what can say amino acids. So therefore that alpha chain is not proper. So hence this in this case, in case if alpha globin production has got few proteins or few amino acids when compared to the required amount, then that causes what we call it as alpha thalassemia. So how this is called? This is mainly the alpha chain, which are two chains. They are controlled by genes HBA1 and HBA2 having 4 alleles, and these genes are present on chromosome 16 of the parent. So if these genes undergo mutation or if these genes are deleted, so then this alpha thalassemia is going to be caused. So now let us come for beta thalassemia. So this is caused due to the two alleles HBB present on chromosome 11. So this also occurs due to mutation of one or both the genes. So for you, alpha and beta thalassemia is important. The third one, which is not there for your syllabus, that is delta thalassemia. It is known to be controlled by a gene called as HBD, and this is also present on chromosome 11, and that forms delta chain of hemoglobin. So therefore an abnormal chain is synthesized and this leads for delta thalassemia. So but children, remember, you should know the difference between alpha and beta thalassemia. So in alpha thalassemia, alpha globin chains, they are not properly synthesized. In beta thalassemia, where the gene, recessive gene is present on chromosome 11, so here the beta chains are not properly synthesized. So this is with regarding to the thalassemia. Now the thing is, see, thalassemia is also a disorder concerned with blood. Sickle cell anemia is also a disorder concerned with blood. What the major difference? So when you if you try to find out the difference between thalassemia and sickle cell anemia, what are the major problem? So in thalassemia, it's a quantitative problem of synthesizing few globin molecules. That's what in case of thalassemia, the major thing is alpha and beta chains of hemoglobin are synthesized in less amount. So therefore we say it's a quantitative problem. Whereas in case of sickle cell anemia, so here it's a qualitative problem because the shape of RBC is known to be affected. Here RBC in case of thalassemia will be normal, but in case of sickle shaped, what happens? The RBC will become sickle cell, and when you compare with normal RBC in a healthy person, you can see the thalassemia RBC, the amount of hemoglobin molecules will be less. So therefore this is the, hence thalassemia is the quantitative problem where less amount of hemoglobin molecules will be synthesized, whereas sickle cell anemia is a qualitative problem. Here the shape of the RBC will be affected. So this is totally with regarding to Mendelian disorders. So with this, all the Mendelian disorders starting from what we can call hemophilia, sickle cell anemia, then you have studied about phenylketonuria, then you have studied about color blindness, and this is the last one, thalassemia. So now we are moving on to the second type of genetic disorder, that is chromosomal disorders. So what is this chromosomal disorders? Already I have told you, these are the disorder caused due to absence or excess or abnormal arrangement of one or more chromosomes. That is, a chromosome part may be deleted or the number of chromosomes may be affected. That is, either addition of chromosome may occur or deletion of chromosomes may occur. So therefore, chromosomal disorder are caused due to absence or excess or abnormal arrangement of one or more chromosome. What are the main cause for this chromosomal disorder? See here, in case of the Mendelian disorder, mutation was the cause. But here, non-disjunction during what we can call cell division results in loss or gain of chromosome. So what is this non-disjunction? You know, you have studied about mitosis and meiosis. So in case of this, there is a stage called as anaphase. So where if it is mitosis, what happens? The chromosome during metaphase, they are arranged at equatorial plane, and during anaphase the chromosome divide along centromere and they move towards opposite pole. But here in case of chromosomal disorder, the movement of chromosome towards opposite pole may not occur properly. That's what we call non-disjunction of chromosome occur. So therefore disjunction is they should separate, but here they will not separate. So what is the cause for this non-disjunction or non-segregation? This is known to occur due to two important conditions or phenomenon. One is what we call it as aneuploidy, and other one is what we call it as polyploidy. So what is aneuploidy? Let us come for this. Aneuploidy, it is the addition or deletion of one or two chromosomes to the original diploid set. Remember, again I am repeating, addition of one or two chromosome or deletion of one or two chromosome from the original diploid set. For example, in man, the diploid set of chromosome is 46. So therefore, in case of aneuploidy, and one chromosome can be added, so chromosome number can become 47, or two chromosome can be added, chromosome number will become 48. Likewise, one chromosome may be deleted, so chromosome number will become 45, or two chromosomes can be deleted, that is chromosome number will become 44. So this addition or deletion of one or two chromosome from the original diploid set is what we call it as aneuploidy, and this aneuploidy is of two types depending upon addition or deletion. So let us come for the two types of aneuploidy. First one is hyperaneuploidy. In this case, addition of one or two chromosomes occurs to the original diploid set. That is, if one chromosome is added, say 2n is diploid, so in man 46, so plus one chromosome is added, so this condition is called as trisomy. So addition of one chromosome to the original diploid set of chromosome is called as trisomy. So the chromosomal disorder caused due to this is Down syndrome and Klinefelter syndrome. The other condition is 2n plus 2, tetrasomy. So here two chromosomes are added to the original diploid set. So there is a condition called as chromosome 18 tetrasomy in human beings, but that is rare. Then second type of aneuploidy is hypoaneuploidy. Deletion of one or two chromosomes from the original diploid set. Again, this is of two types, 2n minus 1, which is called monosomy, and since there is one chromosome absent, the chromosome number becomes 45. The best example for monosomy is Turner syndrome. 2n minus 2, two chromosomes are removed from the original diploid set. So this is called nullisomy. Usually the human beings with this conditions will not survive. So this is with regarding to aneuploidy. Remember, addition or deletion of one or two chromosome to the original diploid set is called aneuploidy. It is of two types, hyperaneuploidy and hypoaneuploidy. Hyperaneuploidy is again of two types, 2n plus 1 trisomy, 2n plus 2 tetrasomy. Hypoaneuploidy is of two types, 2n minus 1 monosomy, 2n minus 2 nullisomy. Now let us come for the second type of reason for what we can call chromosomal disorder is polyploidy. What is this? It's a phenomenon of having more than two sets of chromosome. Here the total set is added. For example, let us say, in man, chromosomes can be haploid or diploid. Haploid is n condition, diploid is 2n. So when chromosomes are added here, total n will be added. For example, if 2n is there, 1 n set will be added, so 3n. Or 2n plus 2n, 4n. 2n plus 3n, 5n. So therefore, one complete set of chromosomes can be added, or one or more than two sets of chromosomes can be present in this case. Such a situation is called as polyploidy. Or you have got another definition. An increase in whole set of chromosome in an organism is called as polyploidy. So the major difference between aneuploidy and polyploidy is, in aneuploidy, only one or two chromosome can be added or deleted. Here it is addition only, no deletion. So usually one complete set of chromosomes will be added. So the main reason for this is failure of cytokinesis. That is, during cell division, after telophase, at telophase, the parental cell will divide into daughter cell. So in this case, what happens? The cytokinesis may not take place in meiosis. As a result, the chromosome sets will increase. So this type of condition of polyploidy is more commonly seen in plants. So polyploidy is not seen in animals, but we can produce in an artificial manner. So this is with regarding to types of what we can call types of ploidy which are known to cause chromosomal disorder. So in the next section we are discussing about chromosomal disorder, and we will start with Down syndrome. Until then, goodbye.